Hello, everyone. I would like to take this opportunity to appreciate the 2020 APRES, the program chair, the president, and many other colleagues to make this virtual meeting possible to allow us to share our research in this pandemic. I hope everyone stays safe and healthy. My talk here is the sequence of Bradyrhizobia strain isolated from peanut nodule. So what is a Bradyrhizobia? Bradyrhizobia is a genus of the soil bacteria containing 41 species. And those species are very common in the soil microbiome, specifically the forest soil and the agriculture soil. This bacterium is like a short rod shape. This is a picture of this bacterium under the electron microscope. And this bacteria grow very slow, about like 8 hours per life cycle, compared with E. coli, which is like 20 minutes per cycle. And this group of bacterium, most of these species, they can form the symbiotic and the nitrogen fixation relationships with different type of legume plants, include some important crops like the soybean, cowpea, and the peanut as well. For example, the Bradyrhizobium diazoefficens that can infect the soybean to form the nodule for nitrogen fixation. And this species, the Bradyrhizobia species, have been under study for more than 10 years already. And since we're working on the peanut nodulation, and we have been looking for the different species, Bradyrhizobia species, in the NCBI to see whether we can identify some genome sequences there. And we found out currently there are 17 Bradyrhizobia strains. We have the public available genome sequences, but those species are mostly identified from crops like soybean and the other as genome species, and even from wheat, but no one actually were isolated from peanuts. So why do we study the Bradyrhizobia in peanut nodule? That is because of the species with the strain, the crop species with the Bradyrhizobia strain, there's some specificity exist for building up the symbiosis relationship. For example, the Bradyrhizobia strain isolated from soybean, if we inoculate the peanut, it cannot form the symbiotic relationship, cannot produce a nodule in peanut roots. And the vice versa, if we isolate the peanut Bradyrhizobia strain to infect the soybean, it cannot form the nodule either. However, the nitrogen fixation efficiency is really a critical phenomena for the sustainable agriculture, for protecting the environment, and also the ecosystem. It is really important for the crop yield as well. With peanut, however, the nitrogen fixation efficiency is relatively low. We can see here with all of those different legume species, the nitrogen fixation efficiency in peanut actually contributes only 55% of the nitrogen needs to for the crop yield to reach up to the full potential. So it leaves us a big potential for us to increase or enhance the nitrogen fixation efficiency in peanut. However, to understand this nitrogen fixation efficiency, we need to understand the triangle between the plant host, the rhizobia, and also the environment. For us, we have been focusing on the peanut, the host, extensively in the past years, and we haven't started with the rhizobia yet. So that is why it is important for us to be able to get the rhizobia, expect the knowledge. So in our lab, we started to isolate the peanut specific Bradyrhizobia strain. What we did, we have the 2 peanut cultivars. One is tip runner, the other is a breeding material, UF487A, with like 1 month old plants, and we dug them out from the field, and then get the 20 individual nodules from each plant, and then we sterilize those nodules and then crush them into the sterile water. And then with the suspension, we script the suspension on the agar, the YEM agar plates. And then we let them grow in the agar plates for 7 days and pick up the single colonies. Those single colonies, based on the size and the color, we picked up like several hundreds of different single colonies. And then we did the colony PCR according to the 16S RNA to amplify the ribosomal RNA. And then we sequence the ribosomal RNA to see the different strains. And from there, we get totally 370 single colonies, the amplicon. And from the sequence, we identified 5 strains. They have different, represent different size and also with different ribosomal RNA sequences, but they all belong to Bradyrhizobia genus. So we get those 5 strains, we inoculate them back to the peanut roots, and of those 5 strains, they can infect the peanut roots and produce the nodules. When we count the nodule number, we find out one of the strain called LB8 actually can form a much higher number of nodules on the peanut roots. And we also observe the growth curve of those 5 different strains. And with LB8 actually is a relatively slow growing strain. And we evaluated the nitrogen fixation efficiency as well. We found out the LB8 actually infected the roots. The nitrogen fixation efficiency is dramatically higher than the several other strains we isolated. So with that, we consider the LB8 as an experimental strain in our lab, and we also get the LB8, the genome, sequenced through the PacBio. So we get the genomic DNA extracted from the LB8 strain and construct the PacBio library, about 20 kilobase inserts, and we sequence those library in 3 different smart cell in the PacBio RS2 system. And from the sequence, we retrieved 377,000 raw sequences in a range of 6 to 40 kilobase, and we assembled those sequences into a single circular chromosome of 8.7 megabase. And we annotated the genome, and the GC content is much higher compared with the plant sequences. We typically get 45 GC content percentage, but for the for this Bradyrhizobia, the GC content is 63%. And the protein-encoding genes we annotated, it was 8,000, more than 8,000 genes. And nearly 5,000 of those genes, we know their function, and some of the other genes, we don't know the function. And the average gene density is about 1 kb per gene. We also identified some non-protein coding gene, about 54. They're not protein coding, but they have the gene structure. And we didn't identify or detect any plasmid sequence. So we further annotated this circular genome, and then we compared with 3 other different public available Bradyrhizobia genomes, the representative ones. Here, the outside circle is a Bradyrhizobia LB8, the peanut strain we isolated, and these strains are with other representative strains comparison. And we noticed in the LB8 genome, there is one region has a higher GC content, much higher GC content region. And that region, we noticed it has 2 clusters of genes family. One is a NIF cluster family, the other is not cluster family. And the NIF cluster family is basically the nitrogen fixation genes, related genes clustered together. And here is another, is not a factor gene families of those genes are clustered together, is related with a nodulation. We also noticed there is another focus I want to point it out is type 3 and the type 4, the secretion system genes we also identified, which means this strain actually may involved with this secretion system. They might involved in the fine-tune of the host specificity. That will be our future focus to understand how the Bradyrhizobia specifically infect the peanut strain. So the further annotation of the LB8 genome, when we compared this genome with the 17 Bradyrhizobia genomes publicly available in ACBI, we found out the sequence identity, the average nucleotide sequence identity ranged from 80 to less than 90%. So basically, the NAI, the average nucleotide identity, is less than 95%, which means this genome is a novel genome, not public available yet. And this genome is mostly closely related with Bradyrhizobia diazoefficens, particularly such as USDA 110, that is isolated from soybean, and compare with other, is less likely related with photosynthetic not gene liking strains, such as the Bradyrhizobia, the strains ORS 278 and the BTI1. And these strains, they're infecting the Genome species. And this infection is a crack entry infection. Previously, we thought it's probably closely related with these strains because for the peanut, it's also using the crack entry infection. And we thought it might be closely related with these 2 strains, but when we get the sequence, it's not. No matter what, the most of the genes related with the fixation genes, their collinear relationship among those strains, they're collinear related. Okay, so we further compared those 4 different strains representative of the 17 different Bradyrhizobia. We found out about like 4,000 of the gene families among like 7,000 to 8,000 gene families. They're common genes. And with the LB8, there are actually 93 gene families. They're unique to the LB8. That is future we will focus on. It will help us to actually understand how the rhizobia, the LB8, the regulatory machineries, how they're contributing to the nitrogen fixation. And also this genome will help us to understand the rhizobia and the host, how they're interacting to each other to contributing to the nitrogen fixation efficiency. Of course, right now we're understanding the rhizobia actually is also contributing to the host stress tolerance. And this genome will help us to understand the interaction of the rhizobia and the host, and also how they're contributing to the host stress tolerance. Furthermore, it can help us to understand how these rhizobia are impacting the soil microbiome composition as well. Initially, what we want to understand is how the crack entry mechanism is happening in the peanut, the Bradyrhizobia infecting the peanut. And this is actually also important because that is the primary infection, the primitive infection methods for the bacteria to the host, which is very different with the other model legume species or other crop legume species using the root hair infection. These mechanisms have not been fully understood yet. And that is our future research focusing. With that, I would like to appreciate our lab members who contribute of the technical part for the bacteria, the isolation, the genome sequencing assembly, and also the annotations. Matthew Crook from the Weber State University and Jean-Michel Ang in the University of Wisconsin helped us with the genome annotation. With that, I also want to appreciate the funding support from the Florida Department of Agriculture and the Consumer Services. And thank you all for your attention to this presentation.